Hey guys, this is Mr. Mahmood, and you are watching Division Lecture 2. In this video, we're going to get into the cell cycle, which is the basic lifespan of one typical cell. Within the cell cycle, there are a few different phases. The phase that most of the cell cycle is spent in is called interphase. So this video is particularly going to focus into interphase within the cell cycle. The last video will get into the last part, which is considered mitosis, the cell division process, and all the detail within it. It's very important to understand basics about DNA before we can really get into this discussion about what the cell cycle is actually like and what occurs within interphase, because DNA, as you hopefully understand by now, is the component that gives all instruction to the cell and is the vital component that without it a cell would not be able to function properly. So when we get into this discussion, keep your understanding of DNA in mind, because we're going to be referring back to it quite a bit. So to get things started, let's think about a basic timeline. If I were to ask you to set up a human timeline, the timeline of somebody that's lived on Earth at some point in the past, your timeline would probably be linear. It would be a straight line. You'd probably have a point on the left at which that person was born, then there would be a straight line in which there's a series of events in that person's life up until a point in which they die. And that's your idea of a timeline for that life, because human life is linear. You're born, you live for a while, you do certain things, and then you die, right? A cell doesn't quite work that way. So instead of thinking about a cell's lifespan as a linear timeline, we're going to think of it as more of a circular timeline. So the process of a cell's life will actually end when new cells are made from that cell, and each of those starts their own lifespan. So it's kind of like a continuous cycle, with the exception of every now and then when cells do break down, they do get damaged, they get destroyed, or they just wear down over a long period of multiple cycles. But most of the time, when we're talking about a cell that's hit the end of its own lifetime, it actually is going to split into two new cells, each of which can turn around and perform the functions of the original cell of the previous generation. So this process of the cell cycle is critically important to the idea of maintaining homeostasis. Cells have to be able to work the exact same way from one generation to the next if you expect an overall organism that's made of multiple cells and multiple tissues and organs and organ systems to be able to work one generation after the next in the same manner without any sort of a change. Every generation of cells that gets made has to be identical. It has to be the exact same type of cell that does the exact same thing and is just as efficient as the one before it. A good reference to think about if you were thinking about two different businesses. Let's say there's one business that has a really high turnover rate. That means people quit a lot and they have to keep hiring new employees over and over again. Every time they hire a new employee, they have to go through the training process and try to get that employee to be as good as the one that was before it. And the likelihood is every single time the new employees probably aren't going to be as good at doing what they're doing because they're inexperienced at it. So every time you get a new round of employees, it's not going to be the same kind of consistency. So that company is probably not going to be very efficient. Whereas if you had a company that has a really low turnover rate, that means people don't really quit very often, they stay for a very long time, the employees become really consistent and really good at what they do, and so that company is going to be a whole lot more efficient. So cells kind of have to work the same way. If cell cycles happen in a certain time frame, when a new cell is developed, or two new cells in this case are developed from one, each of those new cells has to be able to work just as well and just as efficiently as the one before it. And what drives the control of a cell and what drives what a cell is capable of doing is that DNA that's inside of the cell. So it's very important when we start talking about the cell cycle, especially towards the end of this part and into what we're going to talk about in Division Lecture 3, which is the splitting of the cell and making two new cells. It's incredibly important that each of those new cells actually has the exact same genetic information. And the process of making that genetic information happens here in what's called interphase. So interphase is the majority of the cell cycle that you see here. It's the part that's colored in orange. The interphase process is split into three basic segments. The first segment is called G1, or gap 1. That G1 phase actually has something extending off called G0 that some cells fall into. We'll talk more about the G0 in the third lecture. But G1 is the first section. Then it moves into a phase called the S phase, or the synthesis phase. That's very important for this process of making sure the two new cells that get made from this cycle actually have identical DNA. We'll talk about that. And then the third phase is called G2, or gap 2, in which it's sort of final preparations and final components and being prepared so that when the cell does split, not only does the DNA split properly, but everything else splits the same way. So we'll go through one at a time and understand our basic process here. So our starting point is going to be right at the beginning of the G1 phase. So the previous cell cycle has just ended, which means that the original cell has just split into two identical cells, identical to each other and identical to the original cell that had just gone through that cycle. So each of these two new cells will now start their own cycle, and this is where we're going to start off with one of those cells. So the very beginning of the cell cycle is the very beginning of the G1 phase. Now, one of the 8 characteristics of life, which I hope you guys remember, is that all cells grow and develop. I made this joke before. We are fortunate, and your mother especially is very fortunate, that you were significantly smaller when you were born than you are now, right? It definitely made things easier on your mom. So all living things have to grow and develop. We aren't born in our mature form. And even though we more normally think about this more on the large scale, it's actually true at the cellular level as well. Every single cell, when it goes through its cycle, at the very beginning of the cycle, when those cells are created, those cells are actually the smallest that they're going to be within their entire cycle. And over the course of this cycle, as we go through the G1, the S, and the G2, we are going to slowly see an increase of that cell size. And that's actually something that in a human you think of as a good thing. As you get bigger and progress, you become more complex and more advanced at things. But at the cellular level, it's actually not a very good thing. So it's very important to understand that when the cell's starting to grow and develop and getting bigger and bigger, it's actually not working as well as it used to. So at this point, where we're starting, G1, the cell is actually its smallest and its most efficient. So to make a reference to that, let's think about it this way. It has to do with the basic mathematical concept of comparing surface area to the volume of any given structure. When we're talking about the surface area in relation to the volume, the proportion of the two really matters. Let me give you an example. Let's say I was driving across the country, okay, from California up to New York, and I drove the scenic route and I'm going through Texas as well. If I'm driving from far west Texas, like El Paso, to far east Texas, like Tyler or something like that, that distance, compare that distance to, let's say I'm making my way up the East Coast and I'm traveling through Rhode Island from one border to the next. I'm going the same speed all the way through. Which distance, which state do you think I'll be able to pass through more quickly? Texas or Rhode Island? Obviously Rhode Island, right? Because the distance inside of those states is significantly different. So it's related to sort of like the volume within a cell. If I have a really big cell, keep in mind all chemical reactions require certain reactants, and a lot of times those reactants aren't really that close to the part that's needing it. So that means that that specific chemical reaction has to wait for its reactants to get there. And sometimes those reactants actually have to be the products of other chemical reactions, and then they have to travel to that part of the cell. Or maybe they have to come from outside of the cell, pass through the cell membrane through diffusion, and then eventually make it where they're supposed to go. So if I have a really big cell with a whole lot of volume inside, think of like a big state like the state of Texas, to get to any part of the state is going to take a while, and therefore that state has to sit there and wait for all of the different parts, just like a cell has to sit there and wait for all the different reactants to get where they're going. So a really big cell isn't very efficient because it's just waiting for everything to get where it's supposed to go. Whereas if I have a smaller cell, similar in size in terms of the state to like the state of Rhode Island, things can get in and out very quickly because the distance traveled is significantly lower. They're still going in the same rate, they just take a much lower amount of time to get where they're supposed to go. Cells work the same way. When a cell is nice and small, everything can get where it's supposed to go. And when we talk about a cell, it's not quite like being born in terms of an infant is born. When you were an infant, you didn't do much more than drool, drink milk, and dirty your diaper, right? That was about the extent of your capabilities at first. And then as you grew, you became more and more advanced and were able to do more things. A cell doesn't really work like that. When a cell is first made, even though it's very small, it's pretty well developed and it's ready to go. It has all the components needed for it to function normally. So at that G1 phase, it has everything, and it's super small and super efficient. So this is like the time when a cell is really able to work at its highest caliber and its highest level. So this initial part of the G1 phase is very, very important for the cell to do everything it's supposed to do. So what we usually define the G1 phase as is the time frame in which the cell is able to go through its normal functioning. So whatever the cell may be, whatever type of cell it may be, the job that it's supposed to do gets done the most efficiently here in the G1 phase, specifically right at the beginning of the G1 phase. Now over the course of G1, it's going to gradually start getting a little bigger and bigger. And the time frame in which it takes a cell to get through G1 depends on the type of cell. So different cells have different rates of their cycle, and the major differences in their cycle is how much time they spend here in G1. The replication process is pretty much the same in all cells because, as you're going to learn, all cells have the same DNA, so to replicate the same DNA takes about the same amount of time. And the G2 phase is also relatively similar in all different types of cells because final preparation for all of the different cells for division is about the same process. So the only real variation that comes in that cycle in terms of time comes here in this G1 phase. So depending on the type of cell, it takes a longer amount of time to pass through here. For example, the cells of your neurons and your brain cells, you've probably heard this many times before, if you kill a brain cell, you're not going to get them back. That's, depending on who you talk to, pretty much true. Neurons can live anywhere from about 30 years to your entire lifetime. So it's not very common that you see neurons passing through quickly this G1 phase. So they take a long time passing through G1 and actually extend out into this section called G0 for quite a while as well. So it's the time frame that they sit in G1 is very long, and therefore the whole cell cycle is very long. Compare that to, say, the cells of your small intestine that make these little grooves, this tissue that's known as the villi that's responsible for allowing absorption of nutrients into your blood. These cells only last about a day or two in their cycle, which means the G1 phase may only be about 24 hours long. So that process is actually pretty quick. So it's growing so quickly that it's passing through the G1 fairly quickly, and therefore the whole cycle is much faster. So depending on the type of cell, it varies in the time in which it's sitting in G1. Blood cells, for example, last about 4 months, about 120 days through the whole cycle, so about half of that time will probably be sitting in the G1 phase. So the cell is gradually getting bigger and bigger, but while it's in this phase, that's when the cell is able to most efficiently do its job. So most of the time when we think about certain cells that are doing a function for the big organism or for the tissue or the organ system, it's mostly the cells that are in this G1 phase that are actually doing the work that your body needs it to do. As the cell gradually gets to the point where it's ending its G1 phase, it's becoming so large that it's starting to recognize that it's losing its efficiency. And as it recognizes that it's losing its efficiency, the rest of the steps involved in the cycle are all going to be primarily focused on getting that cell ready to divide so that it can start over, get small again, and then jump right back into the beginning of that G1 phase. Before a cell can divide, it has to be sure that it's preparing itself so that when it does split into two new cells, each of those two new cells get everything they need in order to function exactly the same way that the original cell did. And this is a very important component here when we talk about this process of cell division. It's very important that you have this consistency, and that consistency all comes from the information that's responsible for instructing the cell what to do. And as you should know by now, that information is in the form of deoxyribonucleic acid, or DNA. So here's the basic idea. Every single cell in your body originated from one cell. Your first cell was the cell that was the combination of your mother's egg and your father's sperm. That first cell is called the zygote. All right? That zygote contains what we consider the full human DNA line, which is half from your mom, half from your dad, and it combined to make the first full DNA line that made you who you are. That first full DNA line for humans is made up of 46 chromosomes, or if you think about number of bases, it's about 6 billion bases long. So these 46 chromosomes make up your human DNA line. And all of us have 46 chromosomes. The human DNA line is the same from one person to the next. What makes us unique and what makes us different, what makes every cell in your body different, are the parts of your human DNA line that are red. and the different parts that are read will determine different proteins that are made and in turn perform different functions within your cells. But the human DNA line is actually the same. You're pulling the same DNA every single time you're making new cells, and every single time you're making new cells, you have to have the exact same DNA in each one of those cells. So let's just think about this mathematically. You're going to start with that zygote that has 46 chromosomes in its DNA, right? Rather than continuing with this number 46, a lot of times geneticists break down the 46 chromosomes into an algebraic formula where they use the letter N. The letter N represents what each parent contributed in their sperm or their egg. So if their human cell, the whole human DNA line has 46 chromosomes, each parent contributed half of that, right? Mom gave 23 chromosomes, Dad gave 23 chromosomes. We're going to use the letter N in this case. So 46 chromosomes is actually the same as 2N, because remember that's both of your parents contributing together. So let's mathematically think about this. So if we're starting with a 2N cell, that's your zygote. Some other words that we call these 2N cells are diploid. It's di, meaning it's 2N combined together, so that's why it's called a diploid cell. If you ever hear the term diploid cell, we're talking about these body cells that all originated from your zygote, which is also a body cell. Another word for body cell is somatic cell. So these are all words that mean the same thing. So if we hear somatic or body or diploid, these are what we consider the 2N cells, or the 46 chromosome DNA cells. So we're starting with the cell that has 2N. The goal here is that we're going to want to make two new cells that each have the same genetic information as each other and as the parent. So I want to take a 2N cell and I want to make two new cells that each also have 2N. Now just basic math here, something doesn't add up. If I take this 2N cell and I just split it down the middle, I'm not going to get 2N on both sides. I'm just going to get a single N, or just 23 chromosomes on both sides. That would mean I'd make two new cells that now only have half the genetic information as the original, which means they're not going to be identical to the original cell. They're not going to be able to perform all the same functions or make the same cells after it. So something has to happen here in the middle in order to make this work. And that's what this S phase is all about. This synthesis phase is all about making sure that when you split that cell during mitosis and it's split it evenly, you're going to get 2N in one cell, 2N in the other, and they're all identical to each other. So in order for that to happen mathematically, you have to take the original 2N and double it. So something is going to have to happen here to turn the 2N into 4N before the split starts to happen. And that's what the S phase is all about. So S stands for synthesis. Synthesis, as you should remember from dehydration synthesis, means to combine or to come together or to make, right? So the process of the S phase here is all about making two molecules of DNA from one. And it's a very specific process that you're going to have to understand very, very well. But this will ensure that you now have two exact copies of your original DNA line, so that when the cell actually does split during mitosis, one copy goes one way, one copy goes the other way, so that each of the two cells that are created from it are identical to each other and identical to the original. So here's the basic process of how this happens. Let's walk through the basic steps. Here's the diagram that you guys saw from Division Lecture 1. Hopefully you're familiar with this. This is a basic structure of DNA without the helix, without the rotation. We're just showing this to you flat so you can get comfortable with the parts. Remember, you have the three parts of a nucleotide here, a phosphate, a 5-carbon sugar, and then one of 4 nitrogen bases. Remember, Chargaff's rule explains the bases that bond together. A bonds to T, G bonds to C. The hydrogen bonds are what hold the bases together in the middle. Everything else is held together by covalent bonds. Now, the hydrogen bonding here is going to be very important, because you should know covalent bonds are much stronger than hydrogen bonds. So what do you think the benefit of having hydrogen bonds in the middle of your DNA line might be? They're slightly weaker than the covalent bonds, but they're still strong enough to hold things together. The answer comes here, because if I want to actually make two identical copies of this single DNA line, something's going to have to happen. And what you're going to end up doing is using both of the outsides of the original DNA line as your basic template to make your two new copies. So to make your two new copies, you're going to have to open up the DNA line and allow nucleotides to come falling in. And each of the nucleotides will match up on one side as well as on the other. And because of Chargaff's rule, because of the base pairing rules, they'll actually match up mirror images or complementary bases to each other and end up making exactly the same DNA line. So let's walk through exactly how this happens. If I want to start copying the two sides to make my two identical copies, first thing I have to do is open up the DNA, or commonly known as unzip the DNA. And there's a specific enzyme that's going to allow this to happen. The enzyme is represented here by this blue square. It's called helicase. Okay? Helicase is responsible for breaking the hydrogen bonds. And again, it's not that much energy, not nearly as much energy to break a hydrogen bond as it is to break a covalent bond, which is good for your cells because the DNA is often going to be opened up, often going to be unzipped. So it's beneficial that the hydrogen bonding is weaker so that it can actually get open fairly easily. So these enzymes lower the activation energy just enough to get the DNA to actually unzip. So step 1 of DNA replication, helicase will go through and unzip that segment of the DNA line, which means it breaks the hydrogen bonds and allows the two sides of the DNA to separate. Okay, so now I have the DNA unzipped. Step 1 is complete. Now step 2 is going to involve DNA nucleotides coming into the middle of this space so that they can naturally start to match up. Another enzyme is going to lower the activation energy to allow these nucleotides in this space. That enzyme is called DNA polymerase, and it's represented here by the red. Now you need to be specific to calling this DNA polymerase because there's actually another type of polymerase that helps with another process that we'll talk about later. But DNA polymerase will bring DNA nucleotides to the site where it's going to try to bond with its corresponding or complementary bases along each side of the DNA line. So for example, when we have this first DNA nucleotide being brought in, this one is an adenine nucleotide. Just based on the drawing, you can see it's the same color as that green one to the side. So this represents an adenine nucleotide base. It's going to try to match up wherever its corresponding base is based on Chargaff's rule. Now here's a little more detail on DNA that hopefully will make sense to you now that we're going to talk about it. I skipped over this earlier because I think it makes more sense here. You'll notice your DNA line is not actually completely identical from left to right. Let's go back a little. You notice the DNA line is not exactly identical from left to right. Yes, the bases are not the same and they correspond, but if you look on the left side compared to the right side, you see how the left side has a 5 followed by an apostrophe. That represents what's considered the 5' end. And if you look at the right side, it has what's considered the 3' end at the end. So one side of the DNA starts with a 5' end, the other side starts with a 3' end. This all has to do with which of the 5 carbons that make up the sugar is closest to the end of the line. So let's focus in here on the one that's on the left. So here's our 5 carbon sugar. And we're going to sit here and try to count out specifically where the carbons are. The carbons always start the count with whichever one's closest to the base. So this first bend here is the first carbon. This one is carbon 2. This one is carbon 3. This one is carbon 4. And you'll notice how there's actually an oxygen here. So that's not where the 5th carbon is. The 5th carbon actually extends off of the pyramid shape to be the carbon that's here at the top of this bend right here. So these are the 5 carbons of deoxyribose, which is the sugar that's found in DNA. The carbon that's closest to the end of the chain is how you name this being the 5' end. So you see how in this case the 5th carbon is the one that's closest to the end of the chain where the phosphate attaches to. So that's why this one is considered the 5' end. Work your way down this DNA line. And again, we have carbon 1, 2, 3, 4, and 5. In this case, this is the end of the line. So the 3rd carbon is actually the one that's closest to the end on this case. So the 3' end has the 3rd carbon in the sugar closest to the end. The 5' end has the 5th carbon sugar closest to the end. So another way to think about it, if the 5th carbon sugar is closest to the end, then that means the phosphate will bond to it as well. The phosphates of the nucleotide are bonded between an oxygen from that phosphate and that 5th carbon. So if you see a phosphate at the end of the line, that's what we consider the 5' end because it's connected to that 5th carbon closest to the end. If you don't see a phosphate at the end, but you see a sugar at the end, that means that the 3rd carbon is where you're going to try to bond to that previous phosphate. So if there was another nucleotide that came in and bonded here, it would bond to that third carbon, just like you see here. Each of these phosphates bonds to carbon 3. Carbon 3. This is where the phosphate actually bonds. So if you see a sugar at the end of the line, that's the 3' end because it's that third carbon that would be ready to take and accept another phosphate and bring in another nucleotide. If you see a phosphate at the end, that's the 5' end because that means it's already accepted and bonded to the sugar below it. That's part of its own nucleotide. So this is what we consider the 5' end. The reason I spend so much time on this is because I want you to notice how the opposite side of the DNA is actually in the opposite direction. In this case, here's the phosphate, so the whole nucleotide is kind of flipped. So this is carbon 1, here's carbon 2, here's carbon 3, 4, and 5. So you notice again, the third carbon is the closest to the end, and in this case, carbon 5, right here, is the closest to the end because that's where the phosphate attaches, which makes this the 5' end. The reason they're in opposite directions like this is because when the DNA does actually unzip, and the nucleotides start to match up. The nucleotides always match up starting from the 3' end to the 5' end. So let's open up our DNA and now think about what I'm talking about here. Helicase has already gone through and broken the hydrogen bonds and unzipped our DNA line. So we already have the space available, and DNA polymerase is going to start bringing in DNA nucleotides. If both of the DNA lines were actually in the same direction, which meant they both started with the 5' end, or they both started with the 3' end. That means when the nucleotides are coming in to match up, they're going to try to match up to the same end at the same time. So you're going to have way too many nucleotides all in the same place trying to match themselves up. And you can imagine how difficult that might be and how likely errors might occur if they're all trying to match up in the same place. It's very possible that they don't quite match up the way they're supposed to. However, because DNA is opposite, the 5' end on one side is actually the 3' end on the opposite side. One side is copied on one end, while the other side goes all the way to the opposite end to start the process of copying that side. So the DNA nucleotides that are meeting in the middle aren't going to be nearly as confused. One side is going to focus on one end. So for example, in this case, we're going to focus in on the 3' end, which is the one that you see here on the left. So that adenine nucleotide is going to go and start matching up with the first base on the 3' end, which in this case is a thymine. So it's going to draw and match up its bases, the adenine to the thymine. So now we have the first connection of a DNA nucleotide. The next DNA nucleotide that would come in this case, because the blue there represents a guanine on the original line, would be a cytosine, which is the red one there. That cytosine will come in and it'll bond as well. And you'll notice as it bonds with the original DNA line, the side there on the right, those two nucleotides will actually bond together as well. That phosphate will bond to the third carbon of the sugar above it, and now we start seeing a connection of the line. So not only do we have hydrogen bonding reforming here in the middle, but we're also going to have these phosphates forming with the sugars of the other nucleotides to start forming the other side of the DNA line. And this process will continue from the 3' end to the 5' end. The next base will come through. In this case, if I have an adenine, there's the green on the original DNA line. A thymine would be the one nucleotide that would come in and again match up, and again we see the combination combining. Cytosine is the original line, a guanine will come in and match up, and it'll continue to combine. And through this process, you'll see, if you compare this DNA line, the new nucleotide line that's forming here in the middle, to the original DNA side that's there on the left, what do you notice about the base sequence? It's exactly the same, because they're corresponding bases. If I allow this to happen, because of Chargaff's rule, the bases are naturally going to start pairing up so that they're the exact same as what was originally on the other side of the DNA. This is how DNA actually replicates. And then for each of these DNA strands, the third step is going to involve a third enzyme. This one is called ligase, and this is the green square that's there. Ligase is sort of like glue, the way I remember, because glue and ligase both have G and L and things like that, so that's how I remember the enzyme. Ligase is responsible for actually reforming a lot of those bonds and sort of re-gluing everything together, making sure the hydrogen bonds form properly, everything else is structured together, until we end up with this DNA line that's now exactly like its original. And the same thing's going to happen here on the left side. You're going to have DNA polymerase bringing in nucleotides, except this time it starts on the opposite end. It's going to start with the 3' end, which is that guanine at the bottom, and work its way up. As the DNA polymerase brings in nucleotides, ligase is going to form all the gluing together, and now we're going to end up with what's eventually going to be the exact same DNA line here on the other side. So here we are with what originally was just one DNA line, and because of the process of DNA replication, we now have two identical copies of the original DNA, and they happen in opposite ends of each other. The bonding process and the nucleotide bonding happened in opposite ends because the DNA lines are not in the same direction. This is what's known as an anti-parallel formation. The DNA sides, the two sides of the DNA, are anti-parallel. They're exact opposites of each other because one side starts with the 5' end, the other side starts with the 3' end, because it's the 3' end that begins the attachments of these nucleotides. And again, because they're opposite ends, the DNA nucleotides that are going in and attach will only focus in on one side instead of having a whole bunch of nucleotides trying to match up on the same ends at the same time. This makes it much more efficient for the cell, and the DNA is able to replicate with much higher success rate and lower mutation rate. We're now right back to where we started, except we're doubled. We have two of the exact same DNA lines by the time this is all said and done, which means our 2N actually turned into 4N. So instead of having one full line of DNA, we now have two full lines of DNA. So now when it does come time for the cell to split, one copy of the DNA goes into one cell, the other copy goes into the other. So the two cells that are created in the next generation are the exact same as each other, exact same as the cells before it, which is critical because you want to make sure you're always making the exact same cells and you're keeping the cells efficient to maintain homeostasis. So it's all about making exact copies of each other, and that all happens, the DNA copying all happens there in the S phase, or the synthesis phase. And again, this is about the same rate for all cells because all cells have the same DNA line, and it takes about the same time for cells to replicate properly. The final step before we can really go through the division process is to make sure everything else within a cell is ready to split. We've covered the DNA, but understand there's more than just DNA that has to be properly put into each cell in order for the cells to function properly. Eukaryotic cells, for example, need all of their organelles. And we've talked about this before, that not only do cells divide and prepare properly, but there are organelles that have to make sure that there are enough of them made so that when the cell does split, they each have the organelles that they need. We've also mentioned how the mitochondria and the chloroplast, remember we've talked about how they actually divide much like bacteria. They have their own form of division separate from what the cell does. That's what's happening here in the G2 phase, final preparation for mitosis. All of the other organelles are kind of divided and split up and doubled up so that when the cell separates, each one gets what they need. And mitochondria and chloroplasts actually do this entirely on their own. They go through their own division process, while other parts are created specifically by the DNA with extra amounts so that when the cell does split, each new cell gets everything they need to function properly. And again, this is the point where the cell is now grown so much larger that this is the largest and the least efficient that it's ever going to be, especially at the very end of this G2 phase, right where it's about to go into mitosis, is the largest and least efficient it's going to be. It's like driving through the state of Texas. It takes forever for things to get where they go, whereas at the G1 phase, right at the very beginning, it was so small, it was super efficient, everything was going in and out, taking care of itself so well. That's when the cell is doing everything it's supposed to do super efficiently for the body. But now we've gone to this point where the cell is just so big, it's absolutely ready to split. It just needed to make sure it had all the parts ready. And those parts include this thing you see here. It's called a centriole. Centrioles play a very, very important part in the division process when we start talking about mitosis. So we'll talk more about those centrioles later. But all of these steps of interphase are what the cell allows itself to do in terms of normal functioning, and then by the end it's pretty much just a matter of getting ready to split again so that you can start over with two new cells that are each ready to go at the beginning there and most efficient because they're the smallest. I hope you guys are able to follow, and I will see you next time.